Good afternoon, sir. I can continue now, sir.
You ready?
Yes, sir.
Yeah, we are already live. Okay, let's start. Good afternoon, everybody. Welcome to this afternoon episode of Pursue, which is for Pursue 5U, General Pathology with specifics in neoplasia, and we are streaming live from Kolkata. And today we'll be continuing with neoplasia with session 4. And to talk on that, we have Dr. Major Palash Kumar Mandal, who is an associate professor in pathology at Sagore Dutta Medical College, Kolkata, and a lab director at Desun Hospital and Heart Institute, Kolkata. Please keep your mic muted, your camera off, and please don't share your screen. With this, let me request Dr. Mandal, sir, please share your screen and start.
Thank you so much, sir. Good afternoon, everybody. So we continue with our topic on neoplasia. And as you know, neoplasia has been found to have a genetic basis. So the genes are the ones which are most important driving forces for a cancer to happen.
Right, we can see you.
We will be discussing today the molecular basis of cancer, and we will try to elaborate upon some of the genes which are involved in carcinogenesis. We will be discussing it in bits because the whole there are a lot number of genes which are involved in neoplasia, and discussing each one of them individually may not be possible. So we will be restricting our talk to important genes, and we will proceed thereafter. Okay. So as you know, this is a picture which has been taken from Robbins, and this picture I had shown you before also, this chart or the diagram. So flow chart, what it says that normal cell, if it undergoes any damage, there are DNA repair genes who successfully repair the cell and bring back to its normal shape or normal function. However, if there is a failure of DNA repair, or there are inherited mutation in the DNA repair genes, then the mutations in the genome of the somatic cells, they progress, and that progression is further facilitated by the activation of growth promoting genes, inactivation of the tumor suppressor genes, or alteration in the genes that regulate apoptosis. Now, finally, what happens is that there is unregulated cell proliferation or decreased apoptosis and leading to clonal expansion of the damaged cell, which in fact leads to the development of carcinogenesis. Okay, so with this with this knowledge and background, we go back and see what are oncogenes, what are proto-oncogenes, and what are oncoproteins. See, these terminologies are very important. You have to understand what each term means. Don't use it loosely because each has got a separate meaning. Now, what are oncogenes? They are the genes that promote autonomous cell growth in cancer cells. Okay, autonomous cell growth in cancer cells. It is very important. Autonomous, the word autonomous, because there is no further further stimulation which is regulated once the oncogenes become active. Okay, so they are autonomous. They can drive the cell growth. They can promote the cell division by themselves without much external support. So that is the meaning of autonomy. Okay, now what are proto-oncogenes? They are the unmutated cellular counterparts of oncogenes. Unmutated cellular counterparts of oncogenes, and these proto-oncogenes, when they are mutated, they become oncogenes. Okay, so proto means before. Okay, so proto-oncogenes, when they are mutated, they are converted into oncogenes. So normally proto-oncogenes are present in all of us. Okay. Oncoproteins. Oncoproteins are the product of oncogenes. You see, any gene, they will lead to the formation of a protein. Okay. The transcription, the mRNA will lead to formation of protein through the transcription, and that protein leads to further downstream signaling. Okay, that particular protein which has been generated after, which is a product of a particular gene, leads to further downstream signaling, and the whether it is associated with cell cycle, whether it is not associated with cell cycle, there is a continuous signaling which goes on. Okay, so now here we are, since we are discussing oncogenesis, so these oncoproteins are the ones who are the product of oncogenes. Oncogenes are already mutated proto-oncogenes. So the product of mutated proto-oncogenes or the oncogenes, and they do what? They are devoid of internal regulatory mechanism. This is important. Devoid of any internal regulatory mechanism, because normally the proteins which are the product of the proto-oncogenes, they have some internal regulatory mechanisms. There is always a balance. I have told you, there are always checks and bounds for any process in the body. So anything cannot go uninhibited. Okay, so if there is an excessive growth, that growth has to be curtailed or put down. Okay, so similarly, if any untoward signaling happens, there will be some counter signal to stop that unwanted signal which is leading to some action, in this case cell proliferation. Okay, now oncoproteins, they do not have that check to stop their uninhibited growth. So that is a very important point, because that is the crux of the story which leads to the formation of a tumor or tumorogenesis, and their production in the transformed cell does not depend on growth factor or other external signal. Okay, so this is again very important, because they are autonomous. They become autonomous. They do not need any growth factor. Once the oncoproteins are generated, they do not need any further support. They are self-sufficient, and they lead to self-destruction as well. So these are the steps of cell proliferation in a normal cell. What happens in a normal cell? There is a binding of a growth factor to a specific receptor, and activation of the growth factor receptor, which in turn activates several signal transducing proteins in the inner leaflet of plasma membrane. So there are these signal transducing proteins are present in the inner side of plasma membranes. At times, these proteins are seen to have two ends, one protruding outside the plasma membranes and one which is present inside the plasma membrane. Okay, so but the receptor actually is the one which has that particular end, but the protein which is there, which is present in the inner side of the plasma membrane, it is attached to the, it is acted upon by the growth factor receptor, which after activation leads to the signal activation of the signal transducing protein. Am I clear? So these diagrams I had shown you before also, it has very clearly given in Robin. So there is the plasma membrane and there is a growth factor receptor, and the growth factor receptor is usually acted upon by a growth factor which comes from the body, and it is a signal. You see, you can, everything is a signal. So growth factor comes as a signal and attaches itself to the growth factor receptor, meaning that in the cell there has to be some growth. Okay, now this growth factor receptor, what it does, it activates some signal transducing proteins which are present in the inner leaflet of plasma membrane. As I told you, growth factor receptor has got, is a elongated structure, and it has got two ends, one protruding outside. So signal transducing proteins do not have two ends. It is the growth factor receptor which have two ends. So which are, the upper end protrudes outside the cell membrane and where the attachment of these growth factors take place, and the inner part, that gives attachment to some other proteins. Okay, so it activates the signal transduction proteins on the inner leaflet of the plasma membrane, and then what happens? Transmission of this transduced signal across the cytosol, that is the cytoplasm, to the nucleus via second messengers. So it is the first messenger which is happening, that is the signal transduction proteins, and this signal again is transmitted through the cytosol to the nucleus. Why does it have to go to the nucleus? Because nucleus is the area where the cell division will take place. All the cell cycle, everything has to go through the nucleus, because that is the area where the DNA is stored, that is the area where the chromosomes are present. Cytoplasm does not contain DNA, isn't it? So everything has to go to the nucleus. So that is the point of action. Understand? And now the entry and progression of the cell into cell cycle leads to the cell division. Okay. So the second messengers, they carry the signal to the nucleus and lead to cell division. Okay. So these are the names of various proto-oncogenes which have been converted into oncogenes due to by some mutation, and the associated tumors. This is a picture from Robbins, so I don't want to name one by one. So you can see there are some names like this PDGF, this platelet derived growth factor beta chain, leads to development of astrocytoma. FGF, that is fibroblast growth factor, leads to development of stomach cancer. Actually, you see, these are examples. There are multiple mutations which have to happen for a cancer to develop, and not one gene, there are multiple genes also which are involved. Okay, so if you say osteosarcoma, is it only associated with PDGF mutation? No, one of them is PDGF beta mutation. However, there are other mutations as well in any of the tumors. Okay, so that is why a tumor is very difficult to treat. If you can arrest a particular gene or a particular mutation, even then the tumor keeps on progressing if it has acquired the potential of acquired the potential of progression. Okay, so that is very important, and hence you have to ultimately identify a point where everything is converging, and if you can check or limit that point or block that point, then only the tumor progression will actually stop. That is the essence of target therapy or targeted chemotherapy, which which is being propagated these days. Okay, so there are growth factors. I told you growth factors, they are present, as they also come as signals. They also are some proteins which are released from the body itself. So they are the growth factors. They are the growth factor. These are the growth factor receptors. EGF receptor family, epidermal growth factor receptor family. There are all these PDGF receptor families. So these receptor families are attached to the plasma membrane, and the growth factors, they come from through the blood. Okay. So similarly, there are proteins involved in signal transduction. The GTP binding protein, the RAS protein, okay, the BRAF protein, and nuclear regulatory proteins are the MYC proteins. So the mutation in these proteins, they lead to development of various kinds of tumors, as has been stated. And finally, they are the cell cycle regulators, the cyclin, the cyclin-dependent kinases, and so on. Okay, so basically there are a host of genes which are involved at various stages of cell cycle. There are some growth factors, there are some receptors, there are some transducing proteins, regulatory proteins, so on and so forth. So each one of them may be mutated, and each mutation may lead to a product, a particular type of a malignancy. Okay. Now, as it is well understood that growth factors, they stimulate cells to proliferate, but they need to have an attachment. They cannot just go and ask the cell to proliferate by themselves. So the growth factor, each growth factor, suppose a epidermal growth factor is there, so that will only attach to EGFR, that is epidermal growth factor receptor. Similarly, if you have PDGF, that is platelet derived growth factor, it will attach to platelet derived growth factor receptor. So each growth factor will have a specific receptor, then only it can act upon. That is the first primary thing you have to understand. Okay, so a PDGF cannot act on EGFR. Understand? So a cross reaction cannot happen. It has to be very specific. Now most growth factors have a paracrine action. That is, what is that? That is produced by one cell and act on a neighboring cell. Paracrine action. This is has been taught to you in physiology. So most growth factors, that under normal conditions, okay, under normal conditions, all these growth factors have a paracrine action. However, in cancer, they acquire the ability to synthesize the growth factors and self-stimulate. Okay, the cancer cells, they have a ability to synthesize their own growth factors, and the growth factors, not only synthesizing the growth factors, they do have the receptors with them also. So if they produce PDGF, they will have a PDGFR. Okay, so they will self-stimulate. It is a kind of self-stimulation, and this is known as autocrine action. That was paracrine action in a normal cell, and autocrine action in a malignant cell. Okay. Now the two products of other oncogenes like RAS protein or the RAS gene, they are lying along the signal transduction pathways. They cause overexpression of growth factor genes. Okay. So this, what does it mean? This means that there are some oncogenes which are lying across the pathway, which are lying along the pathway, not across, lying along the signal transduction pathway, and by stimulation of a particular oncogene, they may lead to, since they are lying on the pathway itself, they can lead to the production of or the overexpression of a particular growth factor gene. Understand? And which will lead to the secretion of large amount of growth factors. Is it? Have I made it clear? So you see, where does RAS fall? RAS is a signal transducing protein. Okay. So what it does? What it does? It leads to the overexpression of growth factor genes, because growth factors have to come. You need to produce growth factors. Growth factors are present in all of us, because the growth factors have to drive the cell cycle. Growth factor have to attach to the growth factor receptor on a cell and drive the cell cycle. So they are present in all of us, because all of us, the cells are proliferating. Okay. And these special proteins like RAS, what do? They lead to overexpression of growth factor genes because of mutation. Normally that is there, but when it is mutated, that leads to overexpression. This word overexpression is the important, because normal growth factor genes were there, but when it is overexpressed, the cell starts secreting large amount of growth factors. Understand? So this is important. And finally, growth factors contribute to the malignant phenotype by increasing risk of spontaneous or induced mutation in proliferating cell. Because you see, the more there is a theory of Brownian motion, what happens? The molecules which are in motion, they try to collide to each other. So similarly, here also what happens is that the cells which are proliferating more and more, they tend to undergo some mutation. Some mutation. Okay. Excuse me. Yes. So this mutation may be either spontaneous mutation or maybe induced mutation. Okay. So this mutations, the more cell will go into cell cycle, the more proliferation it will take, the chances of mutation are more. Understand? So this is one very basic thing. So that's why the quiescent cell, they have less chance of developing malignancy. The active, the cells which are dividing, actively dividing cells like the mucosal cells, the epithelial cells of the mucous membrane, the endometrial lining, which are continuously shed off, and the cycle is repeated every month. So these cells are more prone to develop mutations. Okay. So this is one very important thing in this proliferation story. Now there are these receptors, growth factor receptors. A class of growth factor receptors are basically the transmembrane proteins, with which I have already told you there is an external binding domain and is a cytoplasmic tyrosine kinase domain. Okay, there are names for these domains. So there is a ligand binding domain. So what does the ligand binding domain do? That attaches the growth factor. And what is the cytoplasmic tyrosine kinase binding domain do? That leads to the activation of the or the phosphorylation of the tyrosine kinase and several substrates in the signaling cascade. Okay, so there is tyrosine phosphorylation. So this tyrosine kinase leads to the phosphorylation of several substrates. Okay, so there is a receptor which is initially in a monomeric form. The receptor dimerizes, means there is a two receptors joined together, they join hands, and eventually leading to some phosphorylation of the several substrates, and this phosphorylation leads to the signaling cascade, and the signaling cascade eventually is carried through the second messengers to the nucleus, where it leads to the cell growth or cell proliferation. Okay, Now the oncogenic versions of these receptors are associated with constitutive dimerization and activation without binding to a growth factor. That is the story. What? That even if there is a no growth, even if there are no growth factors, with these growth factor receptors, if they are mutated, they can lead to dimerization, self-dimerization and activation and further signaling, of course. So that is you have to understand each and every gene, each and every protein which is associated with the cell cycling, with the cell signaling and eventual cell cycling is can itself lead to formation of a tumor and cancer. Now, growth factor receptors can be activated in tumors by different mechanisms like mutations, gene rearrangements, etc. For example, there is a red proto-oncogene which is an oncogene, and it is transformed into an oncogene via mutations and gene rearrangement. Okay, this red proto-oncogene is transformed into an oncogene due to mutations and gene rearrangements. And point mutations in red proto-oncogene leads to main type of multiple endocrine neoplasia types 2A and 2B and familial medullary thyroid carcinoma. Okay, so these are the examples. The proto-oncogene, the red proto-oncogene, what does it lead to if it is mutated? Okay. Now, overexpression of normal forms of growth factor receptors like ERB2 gene, also known as HER2 or NEO, a member of EGFR family, is amplified in approximately 25% of breast cancers and also some adenocarcinomas of ovary, lung, stomach, etc. Okay, the growth factor amplification and mutations have been noted in various type of malignancies, and you see this HER2 neo amplification is very important to detect HER2 neo amplification in breast cancer is very important because we have a drug against this particular HER2 amplification. Okay, so that is called Herceptin. So it blocks the HER2 amplification and reduces the uh, reduces the severity of the breast cancer because HER2 amplification gives a poor prognosis of a breast cancer. Okay. Now coming to signal transducing proteins. So we are coming one by one. Growth factors were there. They may be modified. The growth factor receptors may be modified. Okay. So the growth factor receptors, their modification we discussed, the red proto-oncogene, now or the EGFR. Now the signal transducing protein, the RAS oncogene. So the RAS is a very important oncogene which is a signal transducing protein, and RAS is a member of the small g proteins that bind the guanosine nucleotide, the GTP, guanosine triphosphate, guanosine diphosphate, etc. And it plays an important role in signaling cascade downstream of growth factor receptors. Okay, so growth factor receptors, after being activated by getting a signal from growth factor, that is attachment of growth factor, they lead to this signal transducing, signal transduction, and that one of the signal transduction protein is RAS. There are three RAS genes which have been noted in humans. One is the HRAS, another one is the KRAS, third one is the NRAS. So each of these RAS genes has been found to have been associated with various type of malignancies. Okay, now the mutations in KRAS lead to malignancies in CA colon and pancreas. Okay, mutations in HRAS lead to bladder tumors, whereas mutations in NRAS lead to hemopoietic tumors like leukemias. Okay, so leukemias and lymphomas, to be very specific, lymphomas. Okay, now basically this is, I am going step by step. First we discuss the EGF, the receptor families. Now we are coming to transducing protein family. So what is RAS? How does it act? That is important. You have to understand. You just not the name, knowing the name is not sufficient. You have to know the mode of action also. So where is RAS present? It is attached to the cytoplasmic aspect of the plasma membrane as well as the endoplasmic reticulum and Golgi membrane or the Golgi body. Okay, so this is, you see, this is the growth factor receptor, this is the growth factor, this is the plasma membrane, the three-dimensional picture of a plasma membrane, and this is a binding protein, and this is inactive RAS which is bound to some farnesyl membrane anchor. So it is anchored, it is adhered to the inner aspect of the plasma membrane. This is the inner layer of the plasma membrane. This we know that it's a lipid bilayer, phospholipid bilayer membrane, and the inner aspect is this. The inactive RAS is attached to the inner aspect with this anchor, and it is bound to GDP, guanosine diphosphate. Okay. Now this bridging protein is there. After getting some growth signal, it activates the inactive RAS into active RAS. So inactive RAS, which was bound to the GDP, becomes attached to GTP, guanosine triphosphate, instead of guanosine diphosphate, and this becomes active. Okay, now this active RAS leads to further signaling to the mitogen activated pathway, that MAP kinase pathway. Okay, mitogen activated protein kinase pathway. So these are the ways how a RAS, inactive RAS, becomes active and further leads to the proliferation or transcription in the cell cycle. So once it is activated by growth factor, it normally, what it does, it flips back and forth between an excited signal transmitting state and a quiescent state. Quiescent means this quiet state. Okay. So in inactive state, as I told you, RAS binds to GDP protein, GDP, and in active state it binds to GTP, that is triphosphate. Now activated RAS stimulates the downstream regulators of proliferation, which I told you. Okay, so you have to see that this particular thing leads to the cell cycle. So, sorry because suddenly this screen became blank, maybe due to some disconnection from the power supply. So, what
Fine now.
so what happened? What happens is that this RAS stimulates the downstream regulators of proliferation. So eventually everything has to come down to the nucleus, and the downstream signals, they eventually are the signals for the cell proliferation in the nucleus. Okay, so this has been elaborated by that picture which I showed you just some time back. Just hold on this. Okay, so now there are some, as I told you, everywhere there are some checks and bounds. So in this action of RAS also, there is nucleotide exchange which activates the RAS. Okay, by that.
I think there is some internet issues at your end. Just check that.
Okay, sir.
It was happening earlier also, but it got rectified.
Sir, can you see my screen?
No, I cannot.
Okay.
Can you just join in again? Just come out and then join
Yes,
and then
yes.
present again. Yeah, now please share your screen. Yeah, now it's fine.
Okay, sir.
This was where you stopped, right?
So what happens normally is that there is a cycling which goes on. Cycling means from the active to inactive, inactive to active. So while it is activated, this is catalyzed by this guanine nucleotide releasing proteins. Okay, so releasing proteins are the ones which release the GDP, and they attach the GTP by, and thus activating RAS. Okay, so that is the releasing protein. Now after activation, after the action has been completed, so the RAS has to go back to its normal inactive state. So who does that? That is done by the GTPase activating proteins known as GAPs. Okay, so there are another group of proteins. These are the gaps. You see this yellow capsule like thing is the gap which actually leads to the inactivation from active state to inactive state. Understand? So this is the hydrolysis, and gaps prevent uncontrolled RAS activity. So this is important because everything has to be under control. This is normal cell cycle we are talking about, not the after mutation. Now after mutation what happens? The RAS remains in the active form and leads to continuous cell proliferation. That is the difference. While in normal form, the active RAS, after completing its action, went back to its inactive form again, getting attached with the GDP, whereas in mutated form, that RAS which keeps on attaching itself to the GTP and never goes back to the inactive form, and which will lead to continuous cell proliferation because it is sending signals continuously to the nucleus, and the nucleus will follow the signal and it will keep on proliferating. The cells will keep on proliferating and there will be the uninhibited cell growth. Okay, and after mutation the RAS lies either with a GTP binding pocket or with the enzymatic region. essential for the GTP hydrolysis, markedly reducing the GTPase activity of the RAS protein. Okay, so you see the RAS protein itself has its GTPase action, and that GTPase action, you see, it has a GTPase action which was accelerated by gap. Now this GTPase action is definitely reduced when it is mutated. And in addition, downstream members of the RAS signaling cascade, that is the RAS, the RAF, the MAP kinase, may also be altered in cells, cancer cells, resulting in a similar phenotype, that is the active state of RAS, and that is continuous proliferation of the cell. Okay, so this is RAS. RAS comes as a short note for undergraduate undergraduate examination. So please, whenever you are given a short note, try to discuss about the what is the particular gene. Then you have to draw a diagram. You please practice drawing these type of diagrams with your own hands, and then you have to write about the mode of action, and finally what are the cancers which are associated with a particular protein that has been elaborated here. Okay, so you see RAS is given here, and also we had discussed. Okay, here also we had discussed. So this you have to, as if it is given as a short note, we have to write all these things. Okay, now coming to the next family of tumors, those are the non-receptor tyrosine kinases. Okay, non-receptor tyrosine kinases, the oncogenic mutations in non-receptor associated tyrosine kinases, also which function as a signal transduction pathway and leading to cell growth. So mutations, whenever the mutations are happening, they also lead to the malignant transformation, which are the mutations in non-receptor associated tyrosine kinase. So there was one receptor associated tyrosine kinase, which we gave the example just now. Also another example is EGFR. So EGFR, these are all tyrosine kinase based receptors, and there are some non-receptors. So there are the receptor is not there, but the tyrosine kinase function is there. So the example is that the CABL tyrosine kinase in chronic myeloid leukemia and some acute lymphoblastic leukemias. The ABL gene is translocated from chromosome 9 to chromosome 22, and where it fuses with the BCR gene. In chromosome 22, BCR gene was there or is there, and the ABL gene coming from the chromosome 9 going to the chromosome 22 fuses there, and the resultant chimeric gene constitutively becomes an active BCR-ABL tyrosine kinase the oncogene, and leading to the CML, leading to chronic myeloid leukemia. So this is again taken from Robin. So this is the chromosome 9, which is a long chromosome. This is a chromosome 22, which is a short chromosome. You see, this ABL from the 9th chromosome goes and attaches itself to the short 22 chromosome and forms the hybrid gene or the chimeric gene, the ABL-BCR, and this drives the tyrosine kinase activity, leading to the proliferation in the hemopoietic cells, leading to chronic myelogenous or chronic myeloid leukemia, that is in short CML. Okay, so and the importance of this particular, it is also known as the Philadelphia chromosome, and the treatment of CML has been revolutionized by the drug called imatinib mesylate, which inhibits the BCR-ABL kinase. This is very important because this is the first targeted chemotherapy which was discovered. Okay, so this first chemotherapy was invented, and this targeted chemotherapy led to a number of targeted chemotherapy drugs, therapeutic drugs later on, but this was the first drug to be you can say invented, because the treatment was invented special treatment, that is targeted therapy, and that is acting specifically upon a particular gene or a gene which is a mutated gene. Okay, and there are several other examples also in other myeloproliferative diseases like polycythemia vera. There is point mutation in the tyrosine kinase JAK2 activates transcription factors of the STAT family, which promote the growth factor independent proliferation and survival of tumor cells. Okay, so the mutation in the JAK2 family, which activates the transcription family of STAT, that leads to the proliferation in polycythemia vera. Okay, and also some other myeloproliferative disorders. Mutations in transcription factors. I think I will deal with this and the other one, and I'll leave the rest, the mutations which are the cells tumor suppressor genes, I will not discuss today. So we'll limit our discussion till this point. So transcription factor, that is the one of the proto-oncogene is the make proto-oncogene, and it is expressed in all the eukaryotic cells, and they are required when a quiescent cell receives signal to divide. Okay, and make is involved in carcinogenesis by activating genes like ornithine decarboxylase or cyclin-D2 that are involved in cell cycle proliferation or cell proliferation. And there are various activities of MYC: histone acetylation, reduced cell adhesion, increased telomerase activity, and so on. But the problem is there that there are thousands of MYC binding sites and equal number of genes that may be regulated, and hence a common MYC carcinogenesis pathway or program is difficult to map. So that is why it is easier said that why don't you target a particular binding protein? Why don't you target a mutated protein? It is not so easy, because there are so many binding sites for, especially for this MYC, and so many genes which are activated, that it is very difficult to control or very difficult to pinpoint the source and finally the action, and hence the targeted therapy becomes very difficult in case where the MYC is activated. And while on one hand MYC activation is linked to proliferation, on the other hand there is the cells also undergo apoptosis if MYC activation occurs in absence of growth factors. So if MYC activation is there, growth factors are present, so the cell cycle will proceed, whereas if the growth factors are absent, the cell will undergo apoptosis. So this is indirectly a self-regulatory mechanism. Okay. Now, what tumors are associated with MYC mutations? This is, one is neuroblastoma, that there are two forms, NMYC and LMYC. There is also CMYC also. So this NMYC mutation leads to neuroblastoma. LMYC mutations are found in small cell carcinomas of the lung. Okay. And the MYC regulation, the MYC tumor mutation is seen in Burkitt's lymphoma also. Okay. So in Burkitt lymphoma, which is a translocation which happens in the 8 and 14 chromosomes, these are the normal chromosomes. The MYC oncogene resides here, and it goes from 8 to 14, where Ig gene is located, and by residing with the IgG gene, that is Ig gene, it leads to increased MYC protein. Okay, this increased MYC protein leads to cell proliferation. So that leads to the formation of Burkitt's lymphoma. Cyclins and CDK dependent, and this is the last one actually. Just give me 5 more minutes. I think this is last, because if I don't complete this, it will there will be a lot of things to complete in the next lecture. So cyclins and cyclin-dependent kinases. Eventually, as you know, everything has to home onto the nucleus, and the growth promoting stimuli, the quiescent cell has to enter the cell cycle. Everything, the cell cycle is the one which has to be started. Okay, because if the cell cycle doesn't start, the cell remains in the quiescent stage, there will be no proliferation, and if there is no proliferation, there will be no tumor. Fine. So the cells cancers may grow autonomously if the genes that drive the cell cycle become dysregulated. So the genes which are driving the cell cycle become dysregulated. And what are those genes? They are the two sets of genes are there. One are the CDKs, that is the cyclin dependent kinases, and what are the cyclins. Okay, so the cyclin dependent kinases are activated by binding to cyclins. So these are the CDKs, CDK4, CDK6, CDK2, CDK1, and so on and so forth. So, and they are activated by specific cyclins, like this one is activated by cyclin D, this one is activated by cyclin A, so on and so forth. Okay, and CDK cyclin complexes, what do they do? Sorry, they phosphorylate crucial target proteins. They phosphorylate target proteins and help the cell cycle to progress. Okay, so you see cell cycles we had discussed before also. Just to recapitulate, you have to understand that there are some cell cyclin dependent kinases which are activated by binding to cyclins and inactivated by cyclin dependent kinase inhibitors. So there are inhibitors and there are promoters. Cyclins are the promoters and CDKIs are the inhibitor. This is normal cell cycle. Now what happens? Once the cell cycle progresses, the CDKs they are again deactivated. Okay, and the cyclin levels are rapidly reduced. They are declined. Okay, I mean the levels they decline, and cell cycle a cell cycle is like a relay race. Why it is a relay race? Because you see there are several sets of CDKs and each one is regulated by a different cyclin. So suppose the cyclin D was regulating CDK4 and CDK6, this particular portion CDK2 is regulated by cyclin A. So CDK2 and CDK1 is regulated by cyclin A. Similarly, CDK1 again is regulated by cyclin B. So each step is regulated by a specific cyclin. So it looks like each lap is regulated by a distinct set of cyclins like D, E, A, B, etc. An expression of inhibitors of CDKs is down regulated by mitogenic signal pathway. So in normal condition, there are inhibitors. After a particular action is over, after the target is achieved, the inhibitors come into picture and they dephosphorylate. Okay, so that means the dephosphorylation leads to the inactivation, leads to reduction of the cyclins. Okay, but the if there is a mutation, if there is a mutation, the CDKIs, the inhibitors are down regulated and there is continuous progression of the cell cycle. So this is very obvious. If you think very logically, whenever there is tumor, the inhibitors will be suppressed and the promoters, that is the cyclins, they will be activated. So this is the list again given in Robbins. So list of CDKs and their inhibitors, and there are some checkpoints. A very important one of the checkpoint is a tumor suppressor gene called P53. We will discuss about P53 in our next lecture class. This is a list of tumor suppressor genes, but we will discuss it in the next class. So I think because we have completed our time, and I'm sorry for the interruption which happened in between today and due to some net problems. I have ended my lecture today. Thank you so much.
Thank you, Dr. Palash. Wonderful lecture as usual. Very difficult topic to understand at one go. One really needs to go over and over again to understand this. And this is very essential to understand this particular topic, to understand the various aspects of immunohistochemistry and molecular biology. I think you have really gone in a nice pace to make everybody understand. Very complicated, very difficult. But then I think if people go through your lecture and again and read the book simultaneously, I think they will be able to understand it. Thank you so much.
Yes.
So next time we will have session 5, neoplasia.
Yes, sir.
Yeah.
Yes.
Go slowly, no problem. We have enough.
Thank you, sir.
All right, so take care. Thank you so much.
Okay, sir.
Bye.